Well, thank you very much for coming to my seminar. I'm Duncan Greig, and I'm going to talk to you today about sexual signaling in yeast in Saccharomyces. So, in my group, we do quite a lot of work with Saccharomyces cerevisiae and closely related species. And Saccharomyces is this amazing model system for genetics. And as I'll show you, I hope, it allows us to do really interesting kind of manipulations in order to test evolutionary and perhaps ecological theories. And this image kind of captures, what we'd like to know about yeast. So yeast has this very complex and interesting life cycle, which I'll tell you a bit more about in a minute. But what we don't really know much about is how all of its interesting life cycle and genetics and all its life history and its natural history actually fits into the real world. And I think this is quite a bit of a problem as yeast becomes increasingly sophisticated as a model system for genetics. So we work on lots of different things. And a lot of it is trying to work out how the life cycle fits into nature and how yeast is adapted to its natural environment. And some of the things we do are looking at the microbial communities that yeast exists in, and we also make yeast crosses between different yeast species and do experiments on hybridization and speciation. And the other kind of area that we're focusing on quite a lot is yeast mating, and that's what I'm going to talk about today. And most of the work, so obviously this is pretty informal, so everyone apart from Daniel should feel free to interrupt, because I don't really know how much genetics detail to go into. And it's pretty much open-ended as well. I'll stop talking after 45 minutes or something. So I'm going to start with this guy's work, Dave Rogers, who's a postdoc in my lab. And we might get on to other people's work, but probably it'll mostly be about Dave's work. So I'm thanking him at the beginning because I might not get around to, might not have a wrap-up slide at the end to thank everybody. I would also like to thank, of course, the Newton Institute for having me and all the organisers and making it such a great time for us all, and Thomas in particular, who actually invited me. Okay, so this is a kind of cartoon that represents roughly what we know about the Saccharomyces cerevisiae life cycle. So we think it mostly grows as a diploid, and there are kind of three cell states of Saccharomyces. So they can be diploids, and they're alpha A's, they're heterozygous at the mating type locus, or they can be haploids, and there are two mating types, A and alpha. And we think mostly it grows by mitosis asexually as a diploid organism. So you just get simple cell division, and you probably get some big local expansion of some clone. And then when the resources are exhausted, yeast can do lots of different things. It could just die, and that happens quite a lot. It can form kind of resistant but non-mitotic stages, or it can undergo meiosis and produce 4 haploid spores, which are usually kind of bound together in this thing that's called a tetrad, and they're held together by this ascus, which is the remains of the old diploid cell wall. And that's quite interesting because they tend to remain associated together, so that when they germinate and start become metabolically active again, perhaps when they get food back, very often mating occurs within between the products of a single meiosis. So it's really extreme form of inbreeding of selfing. And what happens is you get the A mating type and the alpha mating type. They mate together, produce new diploids, and the life cycle can continue. And of course, you can get mating between different lineages, potentially, even between different species. You don't necessarily get 4 spores. You sometimes get tetrads with just 2 spores in. It's another interesting characteristic. And yeast can also do this very strange, well, a very unusual type of mating, which is completely within clone mating. So this alpha here, for example, can undergo haploid mitotic division. Then one of the cells can switch mating types, can mate with its clone mate now and make a new diploid, which of course will be completely homozygous across the whole genome. Okay, so what I'm going to talk about is just this process of, in a bit more detail, of the mating process and what signals are necessary for mating to occur. So this cartoon represents the signaling that occurs between A cells and alpha cells. So this A cell produces a sex pheromone. So this is this little blue square, and the A's, it's called A factor or A pheromone. It's excreted into or secreted into the environment by a special membrane transporter. And A cells also express a pheromone from this gene called STE2. Sorry, a pheromone, I beg your pardon, a receptor from this gene called which is called STE2. And A cells don't produce the receptor for their own pheromone, and alpha cells, which produce alpha factor, this is the yellow circle, don't produce a receptor for their own pheromone. So that's important. They don't respond to their own signal. So this is clearly some kind of communication. It's not like an autoinducer, like for a quorum sensing molecule, which could potentially, rather than being a signaling molecule, just be some kind of environmental sensing system. Yeah.
How's that like? Is that all clustered like your receptor for the other guy together with your machinery?
No. So both types have all the genes necessary for all of this stuff. The master switch that determines the mating type is the mating type locus. So a MAT, diploids are heterozygous for A and alpha, and so of course that mating type locus segregates in meiosis, and you'll get two A's and two alphas, and that's just the master switch that determines which genes are expressed. So these could be genetically identical everywhere except at the mating type locus, and they would still have these characteristics. Now the alpha factor is slightly different. It's secreted more like through the normal exocytosis pathway. So it's first expressed as a single protein with multiple repeats of the pheromone peptide sequence sort of in tandem, and I'll talk about in detail a bit more about that later. And then it's processed through the Golgi apparatus and secreted, just like many other secreted proteins. It doesn't have a special transporter. And the alpha factor is hydrophilic and diffuses very readily. The A factor has these modifications to it and is much more hydrophobic. There's one more. So it's a very simple system, right? There are two pheromones and two receptors. But there is one more little interesting thing, and that is that the MAT A cells also express this protease that's secreted outside the cell that degrades the alpha factor. And this is thought to be important for kind of the fine sensing of the factor. But it's not really clear exactly what role it plays. Okay. So the kind of things that we've been interested in to try to determine what sort of information is being transmitted by the pheromones. Why does a system exist? What are they getting from this complicated mating system? So there are really two things you could change about the pheromone. One is the concentration, and the other is the sequence. So these are short peptides. They're 13 amino acids. And so obviously there are many possible sequences, and the concentration can also vary, obviously. And probably one of the most important things is that the information you get from the concentration is you can see how close potential mates are. So if you're choosing between several different sources, if you're mostly attracted to the strongest source, then you'll find a mate more efficiently. That's obviously quite straightforward. But because when the cells sense each other's pheromones, they upregulate their own production of their own pheromone. So there's also this kind of courtship process that goes on where cells become committed to mating at some point. So it also indicates the availability of mates as well. So both cells will upregulate their pheromone production as they become more and more committed to mating. And the mating process is also quite complicated. The cells change shape, they send out projections towards each other, and the projections meet, and then a diploid is formed. And an area that we've been quite interested in is sexual selection and what kind of quality information in terms of mate quality cells might be getting from the pheromone signal. But what I'm going to talk about mostly today is what information they get from the sequence of the peptide. Now, the two sexes, the two mating types, make completely different peptides, so clearly it's important for identifying the right mate, a mate of the right sex. But what I'm going to talk about mostly is about species recognition and the potential role of the peptide sequence in species recognition. So this is just to remind me what I said very briefly earlier. There are the pheromones themselves, both the A pheromone and the alpha pheromone, are both encoded by 2 genes each, and these genes are redundant. So they make the same peptides. So this is MF-alpha-1, it's one of these 2 genes, and as I explained, the pheromone is encoded, in this case, 4 times in tandem in the protein, and then as this protein is processed, these repeat peptide sequences are released and form the mature pheromone. The other gene for the alpha pheromone encodes the same peptide at the second repeat, but the first repeat has a variant peptide which we call alpha prime. Now this one does not vary in the number of repeats. All MF-alpha-2s have just 2 repeats in Saccharomyces cerevisiae, whereas the number of these repeats can vary a lot, as I'll show you if we get to that part. So the first experiment I want to tell you about is we'll just ask how sensitive, how able as Saccharomyces cerevisiae are these receptors, ST3 and ST2, at detecting pheromones from other species. So what we did is we knocked out, let's go back to that. We knocked out all of these 4 repeats, and then we just replaced this last one. So we reduced this gene down to just a single copy of the peptide sequence, and we replaced this sequence with sequences from other species, other yeast species, from both far and close and far away. And we deleted this locus altogether, so there's no pheromone produced from MF-alpha-2. So now our strain is just producing a single pheromone from a single repeat. And we did the same thing for the other mating type. And we tested them and just asked, can this still induce mating? So these results show you whether pheromones are compatible or not. So these are the Saccharomyces pheromones, the 2 alpha pheromones, which I already described to you, and these are the 2 A pheromones. So if they're in bold, it indicates they can induce mating. And of course, their own pheromones can. And these 2 pheromones are sufficient, the 2 different types, to allow mating. And then, so this tree shows lots and lots of different species, and the bold type indicates whether they're compatible. So if they're not bold, they're incompatible and they don't induce mating, or they don't allow any mating that's better than not having any pheromone at all. So this is a really, this is just a cladogram. The branch lengths are not to scale, but I can give you a kind of scale that this kind of amount of divergence is about the same as in the whole vertebrate clade, right? So these are really, really different species. And yet, most of the pheromones can induce mating. So Saccharomyces cerevisiae is not very good at discriminating, identifying species based on their pheromones.
Wait, how? So you've simplified the locus considerably. How good is the simplified locus against relative to the wild?
I can show you that in a sec. I'll just point out one more little thing. This little star here indicates the well-known whole genome duplication in the ancestor. It's also just for scale. So most species, most pheromones that evolved or have a common ancestor since the whole genome duplication are compatible. So to answer your question, Daniel, so this graph shows how good the mating is. So that first thing was just showing whether they can mate at all. This is showing how effectively mating the different pheromones work. So these are the different A pheromones. This is the wild type cerevisiae with no manipulation at all. Okay, and then this is cerevisiae with our modified locus, just expressing the pheromone sequence from MFA1. Right? So it doesn't make any significant difference. This is producing the one from MFA2, doesn't make any difference. This is one from a completely different species, doesn't make any difference. And actually for the A pheromones, most of them either mate very nearly as well as the native pheromones, or not at all. There are only a couple that are kind of intermediate. This is a log scale, so these ones are pretty poor maters. But nevertheless, they still mate, and mate much better than having no pheromone. The alpha pheromones, okay, so this is the wild type as before. This is the normal, the common A pheromone. So for this sequence, there are usually, this is by far the majority component in the pheromone mixture. And it's the one with most of the repeats. And this is that rare variant repeat. And interestingly, that rare one has quite a bit better, induces quite a bit better mating. So Daniel, you can see that both of these are much lower than the wild one. And that's not surprising because the wild type one has 6 repeats in total, and these only have 1. So you get the expected difference. And so here, there's much more variation among different species and how effective their pheromones are at inducing mating. So we were quite interested in why these, and I might come back to this, about why you have these 2 different pheromone types. And so we looked a little bit more closely as we had the system kind of set up. And so this is the data I showed you before. It shows that the rare type, the alpha prime, induces mating more efficiently than the A type. But only at that low pheromone expression level that you get because we've only got a single repeat of each. If you overexpress it by putting it on a multi-copy plasmid, the relationship actually changes around. And so we wondered if this was some kind of, the pheromones have different roles, perhaps at long-range communication and short-range communication. And so we looked at whether how the resistance to BAR1, that protease, differed. And it doesn't actually differ very much, but it does differ significantly. But it was kind of the other way around to what we were expecting. So the common one, the common pheromone, which is not so good at inducing mating at low levels, is also more resistant to BAR1 and not less resistant. So we don't really have even a hand-wavy story for why those 2 types exist. Anyway, back to the main story. So this is rather strange, right? This seems to be a very good, yeast will look the same to me. This is a good way that mates could be chosen and make sure you're not mating with the wrong species. And mating with the wrong species is a really, really bad thing to do. So these Saccharomyces cerevisiae and the ones that share this pheromone, of course, can all mate together. And if they do, the resulting hybrids are sexually sterile. So that's an evolutionary dead end for them. If they mate, they only get to mate once. It's just cell fusion, so that's it. And these ones, where they have the opportunity, potentially, to discriminate, these ones are also either inviable or sterile. So is it just that they never see each other in nature? So there's never been any selection to reinforce the species barrier by having some kind of pre-mating isolation? Or is it that there's some kind of constraint in the signaling system that prevents that from happening? So we did an evolutionary selection experiment to see whether we could evolve just better mating to make discrimination. So we picked the pheromone that was the best, this one, I think. This was the best non-cerevisiae pheromone at inducing mating. So that we'll call that the conspecific, sorry, the heterospecific pheromone. So this is a kind of complicated image that shows what we did, and I'll talk through it slowly. And so we had two different selection treatments, the compatible only one and the mixed one. So this is representing the Saccharomyces, all the cells here are Saccharomyces cerevisiae. They're all the same strain. They're isogenic apart from a few genetic manipulations we made. One of those was to have them express one of two different types of pheromone, the cerevisiae pheromone, which are the ones that are labeled C, or the N-Astelli pheromone, which we've labeled H for heterospecific. So these are alpha cells, and the ones that are labeled ST2 are A cells, and they're expressing, of course, ST2, which is the receptor. So we knocked out the receptor. So the pheromones, again, just as before, they're only expressing the single pheromone from the single locus. Everything else was deleted away. And we also deleted ST2 on its natural locus and put it on a plasmid. And we mutated just the open reading frame of that gene. So here's our wild type ST2, and I kind of represented it by this little Y shape. That's the receptor being expressed on the surface of the cell. And then we mutagenized it, and we did 5 independent mutagenesis pools, 3 at low level and 2 at high level of mutagenesis. So this presumably changes the shape of the receptor in some way. Okay, so we took these pools and then we mated them to the cerevisiae expressing alpha cells. So they're producing the native cerevisiae pheromone. And so they should mate more or less well according to how good their mutated receptors are. And this was our compatible only treatment. And then we repeated this, as I'll show you, through various rounds of evolution. In the mixed treatment, they were mated again in the same way. So this was a 1-to-1 ratio. So we expect reasonably good mating efficiency. So again, we had 1-to-1 ratio just as before, but we added a 9-fold excess of alpha cells expressing that foreign pheromone, the heterospecific pheromone. And the colors indicate genetic markers that we use to select for different types of mating. So in this round, we selected only for cells that mated between the red marker, genetic marker, and the gray. So that we eliminated anything that didn't mate in this treatment. Same here, we selected just for the red and the gray matings. So we eliminated anything that didn't mate, and we also eliminated anything that mated with the wrong species pheromone. So that's like that. We only take the conspecific matings, the cells that mate with a conspecific type, and then we sporulate them again. We then select only the MAT-A cells, the ones that are expressing the pheromone, and then we take these cells and then we make them back to an unevolved population of these, and these, just as before. Okay, so effectively you're constantly back-crossing them to these ones which don't get to evolve. So what we're hoping, of course, is that the receptor, which gets transmitted all the way through here, will be selected according to their mating ability. Okay, that's that. And the other trick that we did to prevent getting resistance to the drugs is that each time we switched around the genetic markers. So in the next round, we select only for the blue-grey matings. That was just a technical trick, really. Okay, we repeated it 5 times, and then we had a look to see what was happening. Okay, so then we did an assay mating. So we mated them under similar types of conditions, but this time we let everything live, regardless of the genetic markers, and then we just determined who mated with whom. So this is how the unevolved receptor mates with the Cerebriciae pheromone, with the conspecific pheromone. So normally you get about 5% mating. So it's not that good, even though there's 50-50 ratio. Without any selection, these are how the different mutated populations mate. And you can see that probably the high mutation rate ones are a bit lower, probably because more of the receptors are completely messed up. And then after 5 rounds of selection, in the conspecific treatment, the mating efficiency has increased quite a lot, so that's good. So there's mutations in the receptors that improve mating being selected. And the same thing is happening in the mixed treatment that consists of the conspecific and the heterospecific pheromone, but we, of course, only allow the conspecific matings to make it through selection. Okay, so then we tested in the future images, the filled bars show the mating rate with the conspecific pheromone. So then we tested them with the heterospecific pheromone. So more or less the same thing with the unselected ones. Again, you can compare it to the unmutated receptor. Now, in the conspecific only ones, these have never seen this heterospecific pheromone, and yet the mating efficiency has increased quite a bit. So this is suggesting that you're getting mutations that simply just, they're more sensitive receptors, they're just increasing mating generally. In the mixed one, though, they've also increased, but nowhere near as much. So this is suggesting then that there has been some selection against mating with this pheromone, which is what we imposed. But what, of course, we really want to see is what happens when we mix the two together. So that's the next slide. So now the bars show the total mating efficiency, but the filled part shows the mating with the conspecific pheromone, and the open part shows the mating with the heterospecific pheromone. So in the conspecific treatment, where they never saw the heterospecific pheromone, as we suspected, as well as increasing efficiency with the one they were selected for, they've also increased mating efficiency even more with the one that they'd never even seen. So again, this just suggests you're getting mutations that increase mating efficiency generally. But with the mixed ones, where we selected against mating with a heterospecific pheromone, this part of the bar is much smaller. And just by looking at this ratio, we can determine a preference for one, or a discrimination measure for one type over the other. So interestingly, in the conspecific ones, they actually discriminate less well. So selecting for improved mating is also selected for less specificity. Whereas in the mixed ones, we got this really nice increase in discrimination. So, perhaps 80 times discrimination. So there's an 80-fold preference for now the conspecific pheromone over the heterospecific pheromone. So it seems to have responded for selection as we hoped. This is just to verify that the mutations were not mutations elsewhere in the genome, they really were in the pheromones. So we just in the receptors, we extracted the pools of plasmid, transformed them back in to the unmutated ancestor, and tested them again, and it looks exactly the same as the last graph that I showed you. So then we started digging down and looking at the individual mutants, right? So we took those individually evolved receptors and tested them separately. So here's a sample. These are the ones from the compatible only mating selection treatment. So some of them, this is the unmutated ST Okay, so again, the bars are the efficiency, the circles are the discrimination measure, the preference measure. So some of them haven't really changed very much, but some of them have really big increases in mating efficiency and as we saw before, increases in mating efficiency, even with the one that they've never seen. So in this case, just a single mutation can do that. With the ones that were evolved to be discriminating, we see these nice big increases in discrimination and big increases in efficiency. But we don't see any single mutations here. Most of these have multiple mutations. And I'll show you what those different mutations do on the slide after this. But this is also kind of interesting. So we took 3 of these plasmids, so these mutant receptors, well, that's the non-mutated receptor, the black one, and these 2, which both showed good discrimination and good mating efficiency. And we tested them against, against more pheromones that they'd never seen, okay? So again, this is discrimination, but now it's discrimination not against between the cerevisiae pheromone and the one that we the pheromone that we tested, but against pheromones that they'd never seen before. So, well, actually, this is the control one. So those are the same as those points. Now this is that variant cerevisiae one. So the unmutated one, has a moderate preference for that, but these ones have actually discriminate against now this the variant alpha factor. This is a different pheromone, and again, you can see that these 2 evolved ones have much higher discrimination against than the native, the unmutated one. And that's not necessarily true for all of them, but in general, it appears that discrimination has increased even against pheromones that they weren't selected against. So this is what happens if we pick out these, separate all these individual mutations and see what happens. So again, this is the discrimination, this is the mating efficiency. And you see this really, and in the middle here is the unmutated receptor. So it has some level of mating efficiency, some level of discrimination. And then mutations either single mutations either increase the discrimination and decrease the efficiency, or they increase the efficiency and decrease the discrimination. And it really is as nice as that. So that middle one's the ST2, so this is now just normalized against, standardized against the unmutated receptor. And you see this amazing relationship between the strong trade-off between the ability to discriminate and the ability to mate efficiently. So you can only get high, both efficient and discriminating receptors if you get 2 mutations in a receptor. Okay, this is showing the structure of the receptor and just how the mutations map on. And I don't think I'm going to talk about this because I think we should move on to something slightly different. So just to sum up this part, so these results basically show that the natural receptor is very poor at discriminating between different species, but that in the lab we can easily evolve discrimination. So something strange is happening. So either they're just not seeing each other in nature, so there's no selection, or perhaps the adaptive landscape, as I've suggested, because of this trade-off between the ability to discriminate and the ability to mate well, prevents mutations from occurring, because you'd have to take 2 steps on the adaptive landscape in order to get both discrimination and highly efficient mating.
So there is just one question about that. So you do see there is some discrimination for wild type, but maybe it's enough in practice to stop them. If you just put 3, so you're not sort of competing them in your scenarios. But if you put 3 on a
in the evolution experiment, they were competing, right?
But no, but I mean, you're asking discrimination where you just have two and you ask how often they get together, but you're not allowing them the choice.
Yeah, we are. So,
Okay.
so those first two, that one's without choice, right? That's just how well they mate with the Cerevisia pheromone. That one is without choice. That's how well they mate with the Castelli pheromone. That's choice. They're mixed together, and you're asking how many mate
Okay.
with them. So they're competing in this case. So these conditions are just like the evolution experiment conditions, right? This assay condition. So in the evolution experiment, they also competed. But I mean, what you're saying in nature might be true. I mean, if yeast usually mate within the tetrad anyway, and usually self, then there aren't that many opportunities, to mate with another species, quite possibly.
Yeah. Well, there's some other feature of the environment which makes it more discriminating.
Yeah, I think it's quite hard to. You can't see them, right? They're not like animals that you can see what they're actually doing. Okay, so I was just talking about this kind of part of it, about how the sequence affects the mating behavior. And now I'm going to talk a little bit more about how the concentration determines the mating. So as I mentioned, if you ask a yeast geneticist, what's the structure of MF alpha 1, they'll say that it has these 4 repeats, but this is very typical of yeast, that most people study just a single strain. And actually, if you look at natural strains, you see really great variation in the number of pheromone repeats. And given how important mating is, it seems a bit strange that there's all this variation. So, well, we had a look at this in natural strains to see how it varied and see if we could figure out a bit more about why it varied. So we built a tree of the regions that flank this gene, MF alpha 1. And then we, in each MF alpha 1 gene, we counted how many repeats there were. So these are the data for different Saccharomyces cerevisiae strains, and these for different Saccharomyces paradoxus strains. So cerevisiae is what we all know about, but paradoxus is a very close relative, but it's only found in the wild. It's not found in fermentation. So all of these strains actually were isolated from oak trees, from oak tree bark. So it's a bit hard to see these faint colours. Sorry about that. So here are the letter tells you what the nucleotide sequences, the nucleotide sequence identity. So of course the amino acid and the amino acids in the peptide don't vary at all, but there are non-synonymous differences, synonymous differences in their nucleotides. So we can identify different repeats. So this one, for example, has 5 repeats, and these middle 2 CC are identical. Okay, so you see quite a lot of variation, even among quite closely related strains. But what you tend to see is that the middle repeats seem to be the ones that are changing, because they tend to be more identical than the ones at the ends. Okay, and that's true also for the paradoxus ones too. So this is for MF alpha 1, though the gene has variation in the repeat number. We also took a look at how MF alpha 1 and MF alpha 2 differed in their kind of molecular evolution. So this is just a reminder that MF alpha 2 has the same peptide sequence for the second repeat, but this variation, this variant in the first one. So we just tried taking all of the nucleotide sequences for all of the repeats and then drawing a tree out of that. And what you see, what you tend to see that happens is that for MF alpha 2, so these are all for various different species, and these would have been a very busy tree, so we just collapsed these clades into one head. So what you see is that for MF alpha 2, the first repeat and the second repeat are more closely related to the first repeat in different species than they are to the second repeat in the same species. Whereas for MF alpha 1, that's not true, of course, because we see this, apparently this rapid copying of, so for the, yeah, so you basically just get this, oh, now I'm having a hard time interpreting my own image here. Okay, I think I'll just move on because I can't remember what the point of this was. Sorry. Oh, good, I've got a summary slide. Okay, good. So for MF alpha 1, there's a lot of variation, and the repeat similarity is they're very similar, suggesting that they're often just copying. You get this sort of concerted evolution. You're getting increases and decreases of copy number. Whereas MF alpha 2, they're clearly very different from each other. In fact, they're so different from each other that they're more similar. The first repeat is more similar to the first repeat in other species than it is to the second repeat. So what effect does it have, this variation in repeat number? Well, it has a big effect on the amount of pheromone that's actually produced. So this graph shows all these same strains that I showed you before for F's paradoxus, and these are all isolated from oak trees, so they all came from the same environment. And at this end of the graph, we've got the ones that just have 2 repeats, and at this end of the graph, we've got the ones that have got the maximum number of repeats, which in paradoxus, in this case, it's 4. So there's quite a lot of variation, and there appears to be some sort of relationship that the ones that have more repeats actually make more. Now the 2 bars here are just showing the constitutive and induced levels of expression. As I said, you get this communication between the 2 cells, so when they sense each other, they upregulate their own pheromone expression. Now, of course, we can't tell much from this, because these are all different strains, so they might be different for all kinds of reasons. Might have nothing to do with the sequence of the gene encoding the pheromone itself. So we took these representations of these 3 different gene types, or 4 in this case, and put them into the same strain. And now again, we've got this is the induced, this is the constitutive level, and so the bar just shows the mean of that. And that's the amount of pheromone that's being secreted, which clearly increases as you increase the repeat number. And then this shows that this actually translates to something biological. If you make more pheromone, you mate better, right? So in competitive mating assays, I think we standardized against this one. If you have an extra repeat, then you actually mate better. So this is important, right? If strains were competing, if you can make more pheromone, you should be more reproductively successful. So, but in this case, we just took the genes out of these different strains and put them into yeast. What if everything is exactly the same, and the only thing we change is the repeat number? So now the sequences are exactly the same. We engineer different numbers of repeats. And by doing this, we can really exaggerate the natural range, going from 1 to 8. So here you see something quite interesting. You see the amount of pheromone that's being produced plateaus out. In fact, it possibly actually drops off. So again, this is the induced, and this is the constitutive, and the bar showing the mean. And the same is also true of the mating success. So having 8 repeats is actually worse for you for mating than having less than that, which is rather strange, because this should be a great way to make more pheromone. So we're trying to figure out some sort of mechanism for why this is, why having more copies should actually make you less good at mating. It's not because of transcription. So having a longer gene with more repeats in doesn't affect the amount of transcript that's produced. Well, it may affect it, but it's not true that having more gives you less transcript. And it's not that the secretion apparatus is overwhelmed. Because if we overexpress MF alpha 1, then on a high copy plasmid, then you could get loads more pheromone secretion. So it must be something to do with the protein translation, that longer transcripts somehow aren't translated as well. And this fits with some other kind of protein translation studies that have come out recently. So I'm going to wrap up now because it's almost time. So I just want to very briefly just say one more thing about why this is interesting and why pheromone signaling is quite important from an evolutionary biology point of view. Making the pheromone has a very high metabolic cost. And we think that this might, this could act as a handicap signal in classical sort of sexual selection theory. And the idea of the handicap principle is that it explains the evolution of sexual displays because as long as the display has some kind of viability cost, then it can act as an honest signal of mate quality, usually male quality. So males that have a large tail, if they're peacocks, which is of course a cliche that everyone thinks about, are attracted to females because the female can tell from this that their male must be good quality because he can afford to carry this very costly display. And so one of the first things we did that got us into this was we did this experiment where we manipulated the genetic quality of had different genetic backgrounds, and then we knocked out the pheromone signal altogether, so they weren't making any pheromone signal, and then we asked how much better could they grow. And so this is showing the relative fitness of the non-producer over the producer, and fitness here is asexual fitness. Clearly, it's not sexual fitness. They wouldn't be able to even mate if they don't produce any pheromone. But they can grow perhaps 30% faster if they don't have the cost of producing this pheromone signal. So this has the characteristics that you'd expect of a handicap, of an honest sexual display. It's cheaper for high quality individuals to produce the signal than it is for low quality individuals. So that was the kind of motivation for looking more closely at the other pheromone signal. Okay, I'm not going to go on anymore. But yeah, okay, that's it for now.
So I'm just thinking about all the genes that are expressed at a particular time and the proteins that are produced. Like how much, if it's really this costly to produce that protein, like how much of the total protein of the cell is it? It seems like it would be a drop in the bucket for an
No, it's not. It's actually quite. So if you just go back to one of these ones, this is telling you, I think the scale is, yeah, this is molecules per cell per second, right? It's making 3,000 of those little peptide molecules every second. It's quite a lot.
Like
so it's hard to compare that to the total protein production, but the estimates are around about 1%, right? So it's nowhere near 30%. So that, just adding up the proteins doesn't work like that. But it is a lot, and they're really pumping it out. Okay, thanks.